Yes. So today we have our April voice talk with Pavel Linhart. And as usual, hello. And Pavel, I'm very excited because Pavel will take us to the world of zoology, which isn't somewhere we've been before, I think, at the Center of Voice Studies. And Pavel, please correct me if this is incorrect. Pavel is now based at the Department of Zoology at the University of South Bohemia in the Czech Republic, where you are currently working on your grant project, which focuses on bioacoustic AI for wildlife protection. That's correct.
It's also
Yes.
one of the
One of the projects. And Pavel had various grants that took him to, I'm going to read my notes now, intrasexual selection drive, evolution of individual identity in animal signaling. Yes, also you had projects on the evolution of identity. You have worked in the areas of behavioral ecology, animal vocal communication, acoustic monitoring of animals. I'm sure there are grants that I'm omitting. Pavel has been based in different places. He's worked, well, right now he's actually in Denmark. He is also at the same time simultaneously in the Czech Republic. Pavel also worked in Poland and has done research work in South Africa, Hungary, the US, the UK, Madagascar, Germany as well, I think. No, not Germany. Various countries across the globe. And Pavel has worked on birds a lot, I know from our conversations, and from your publications, I could see that you have worked on chiffchaffs, willow warblers, tree pipits, and then also wolves, piglets, gibbons, and there are probably many more animal species that we will hear about today. So we are very excited. Take it away.
Okay, thank you, Misha, for a nice introduction. Good afternoon, everyone. So I will start the screen sharing, and I will start with the presentation. So share screen. I will share also the sound. There will be, as example, not my own, but there will be some sounds of elephants, very deep sounds. So maybe if you have headphones, it's good to wear them to hear it. And I'm sharing the screen now. No. Okay. And this is presentation. So I hope you see the presentation now. Can you confirm?
We can see your presenter view.
Yeah.
And now the presentation itself, yes.
Yeah. Cool. So the talk is called Look Who's Talking, and also from what Misha said, it's apparent that my main research interest now is on individual identity signaling, individual recognition. And I will give you a brief overview what you would hear today. So I thought that because you're mainly people interested in human animals, it would be maybe good idea to give you some broad overview of more topics. So first, I will talk very general about my own motivation to study individual identification. I will also I will present how typical study look like in this area, because you may be familiar with how to study voice in humans, but maybe it's a little bit different for different animals. Then I will present my own research on measuring and comparing individual identity in vocalizations. And when we started to measure and compare, then we started to ask what drives the evolution of individuality in vocalizations. And at the end, I hope we will manage in time. There is a section about humans and maybe what is different, what is similar, what is different when we look at humans and other animals. So my first interest in vocal individuality started during my PhD studies, and it concerned the two small songbird species, chiffchaff, which is krænsanger in Danish, and willow warble, which is lavsanger. They are tiny birds with very nice and significant song. So I will play it. I hope you will hear. So this is chiffchaff. And this is willow warble. You can already hear chiffchaffs outside. They already arrived from North Africa to breed, and soon you will also be able to hear willow warbles. They are a very common species in Czechia, basically all around the Europe, in Denmark as well. So for my research, I needed to catch them and mark them. So there is good reasons why you need to know who are the individuals that you study, and for that purpose we catch them and we put the rings on them. Either it can be aluminum ring or a combination of color rings or both. So the color rings allow you to know the identity without recapturing the birds, because you just need to use binoculars and read the color combination. So I think millions of birds are captured per year, and this is for studying many ecological questions. In my case, it was I needed to do behavioral experiment with individuals, and I needed to be sure that I'm not testing the same individual twice. So after catching, after measuring the birds, and I would ring them, and then I would move to another bird. So what I realized that when we catch the birds, we do it like we go into the territory of the bird, we set up the mist net, and then we try to lure them into the mist net using playback. So if you start the playback of the song of the bird in the territory, they see it as a threat, and they try to chase off the intruder. And usually with willow warblers and with chiffchaffs, this is very easy. If you start a playback, usually you can get the bird within few minutes. Basically all males that I attempted to capture were caught after 3 to 5 minutes. But what happened next time, once you catch the bird, you put the ring on and you try to catch the same bird again, this is actually very difficult. So, and it doesn't need to be just next week or next month, but it appears that even like after in the second year, when they're coming back again from wintering grounds, so if we had birds that were already ringed, we were not able even to catch 60% of those males even after 10 minutes. And that was our time when we finished the catching. So at that time, I started to think that it would be very nice to have some non-invasive monitoring technique of individuals so that we don't have to capture them. It would be nice to have such methods for different reasons, because sometimes it's rare, but it can happen that the rings can cause some injuries or health problems. In some habitats or with some species, it's difficult to catch the birds even for the first time. And in our case, it was the problem that when we catch the birds, we change their behavior, and then we can't use them for more playback experiments because they just behave differently. So I started to ask whether it is possible to identify birds from their songs. And I started to read the studies, and almost so there was many studies looking at vocal individuality in birds, in mammals, and often they were also connected with some claims about potential applications for conservation, so that we can use the individuality in voice to monitor the individuals non-invasively. At the same time, most of the studies looked a bit incomplete. They looked like byproduct studies of some other research. And why I'm saying this? So, for example, how does typical study look like in vocal individuality and animal vocalization? How does it look like? So first of all, the sampling is not so great as you might expect. Usually, so you can save your efforts while doing recording by collecting the recordings in the field. So on average, the study would use about 10 different individuals and 15 calls per one individual. So that's not so much recording in field. And if you are focusing on some charismatic species, you can even get by with some really modest sample size, like 3 individuals and 8 calls per individual. And still you can publish in some quite respected journals, which is interesting. So let's see. Let's see some example. So let's pretend, for example, that we are going to do the research on elephants. And I downloaded some examples from Elephant Ethogram. These are 3 individuals, Enid, Echo, and Eudora, and I will play you the recordings. Did you hear the chiff-chaff and willow warbler?
Yes.
So yes, good. So I can, I'm not saying something and you don't actually and you can't hear it. So I will play you the 3 examples and listen differences. And these will be deep sound, so sometimes laptops cannot transmit well. So that was Enid, Echo. And Eudora. So now we recorded the calls in field or in zoo or somewhere else, and now we need to analyze our recordings. So again, what you do, you measure the calls, and different studies, different species use different set of measurements, but on average, the studies use about 11 different variables, which can be so they use different sets of measurements, but as with humans, what we are looking at is temporal measurements, durations, intervals between some subunits. We focus on frequency. It could be like modulation of peak frequency, modulation of fundamental frequency, and it could be also the sound quality. That means some assessment of formants, harmonics, assessment of distribution of spectrum, and so on. So we measure the call, and then we go on with further analysis. And usually the studies use this. Usually the studies used discrimination analysis, and nowadays it's more also about other classification, more modern classification methods, with using artificial intelligence, for example. But the principle is the same. So you have the samples, you construct some discrimination model, and then you ask the model to assign the calls to the speaker or to the individual that made them. And if you are lucky enough, or if you focus on good species, you can get very nice. So let's see what is in the graph. So x-axis is duration, y-axis is peak frequency. And echo, for example, if echo has all the calls which are long and high frequency, something like that. If all the calls are the same, they will appear on the graph on the top right. And Eudora could be low frequency and short, like something like that. So if there is like high consistency of calls within individual and at the same time there are big differences between them, we get nice clustering and we get good discrimination success, good discrimination accuracy. And on the left now you will see the case when the individuality is low, and in that case the calls of two individuals overlap and it's very difficult to assign them to the individual using any kind of method. So this is how did it look like in one of the studies on elephants. And usually, so this is usually the picture that you can see. So there are some individuals that that are very different, that are unique, and there are some individuals that overlap with their features with other individuals as well. So this is more one of the outputs of discrimination analysis or other classification methods would be the confusion matrix. And again, if there is high individuality in calls, you will get nice discrimination and high accuracy of discrimination. So the calls are assigned to correct individuals in high percentage of cases. So then the typical study would go to conclusions, and usual conclusions are: we found that calls of, for example, African elephants are individually distinct. And this is the results that this is the result that is almost always guaranteed. There is very few animal species for which it doesn't hold true. Very frequently, there is also the other conclusion that there is potential to use the calls for individual identification and use it for conservation, ecology, other ecological purposes. And quite often, this is very optimistic, and there is not much skepticism about this. So it might look like there's many species in which it's very easy to distinguish individuals and use acoustic monitoring. So then you can publish the paper and you can go on to another call. So we have species that have different call types. So they might use 3, 4, 5 different call types. So in the elephants, it was rumble call, but they have also other types of calls. So you can do this on different calls. You can go to different population. For example, in elephants, it has been done in wild populations or in captive populations. You can look on different sexes, so whether males, females have different individuality in their calls, or you can just go, for example, if you travel, where you go to another country, you can record another species and work on another species. So this is kind of a typical study on individual identity in animals. But so when I was reading this, I was it's on one hand, this is fascinating to see that that it works in a similar way, like in humans. We have good experience with our voices, that we recognize different individuals by their voice. So people always ask about whether animals can do that too. So almost always they can. But it's still fascinating. What is a bit frustrating is that there is same pattern over the years. So it looks like that the studies remain on most of the studies remain on this descriptive part of spectrum, and they are superficial and they don't go too deep, and they don't ask more complicated questions, such as whether actually when we find individuality, whether it's high or low, what is enough individuality in call, what is enough to to allow safe recognition. Whether, for example, individuality differs in males and females, like basically doing these comparisons between different situations, comparisons between different species. So like what is missing in general is accumulating the information and getting some conclusions from a huge amount of studies that have been published. So that's why I was thinking that we should do something about that, and we should look at how individuality is measured and how can we compare it, and whether we can do this actually. I think it's apparent that different species have different level of individuality. So this is example which I borrowed from visual identity signals, and you see 4 individuals of Polistes fuscatus. And you can see that it's might be very easy to distinguish those individuals because they have colorful patterns on their heads, and the researchers actually call it like wasp. They call it faces, because it's very similar to what we see on human faces. But this is not the case for all the species. So this is Polistes metricus, and in that species, it's very difficult actually. So they lack the colorful patterns, and it's not so easy to distinguish the individuals. And so on the first side, we can somehow see that the level of individuality is different in those 2 species. And actually, Polistes fuscatus is a species which has higher social complexity, and they probably need better individual recognition. But also, we can test the animals about their discrimination abilities, and it seems like Polistes fuscatus, they have better signals to advertise their individuality, but they are also better in recognizing, and they are better in recognizing their own species, but also the other species, which is very interesting. But the question is how can we measure this? How can we measure individuality? So if we measure distance, we can do it with different tools. We can do it with different methods. We can compare distance to different objects. So I just listed 4 of them, and if we want to compare them, we would need to have them on the same scale. And nowadays, most of the countries use the metric system. So what we would do, we would convert them all in the metric system. And then we could do the comparisons. So with identity, people introduced different ways how to measure individual identity. So one of the first was just doing the ANOVA with individual as a factor to see, and then you can use the F value as kind of, or effect size as kind of measure of individuality, how pronounced it is. Then also discriminating function analysis has been introduced quite early, and it remained the dominant method for a long time. But there are also other, like during the time, researchers were coming with other metrics. One of them is potential of individual coding. Another based on information theory is Beecher's information statistic, which is expressed in bits. Signature efficiency, also based in information theory, again in bits, but but calculated in different way. And mutual information, the same, again, like expressed in bits. So, but we don't have the metric system for those values, and we don't know how to compare them, how to put them on the same scale. So that's what we tried in our study, where we did a review of individuality metrics, and where we used the simulated data sets, and we used some real data sets, and we tested the performance of the individuality metrics, individuality measures, and how they behave in different situations. And we tried to find out which one is the best one, or which one is actually well suited for comparisons, and which performs the best, and what is the relation between them. How can we convert between them? So I will not go into the details. So I will just skip to the conclusions. Identity metric that we have, and that's because some of the measures, one important drawbacks of, for example, discrimination analysis, is that it expresses a bias. The more individuals you have in your study, the lower will be, like in general, the lower will be the discrimination accuracy. It's because the more individuals you have, the more likely it is that you will find two individuals that are very similar, for example, and you will get overlap and you will get confusion. So this is the bias. If you measure large population, many individuals, you will get lower discrimination, and that's not what you expect from a good measure. So another finding was that actually people are duplicating the metrics, so they use different metrics with different calculation, but actually they are very well correlated, and they can even use those in the same study, like two metrics of individuality. And as a last point, so we actually provided tools to convert between discrimination analysis to Beecher statistic. So based on how many individuals, how many calls you had in the study, and what was the discrimination score or discrimination accuracy, based on this information, we were able to convert to Beecher's statistic on the same scale to make comparisons between different studies. So just to give you an idea about what the Beecher statistic Beecher's information statistic is, it's very easy in principle. You can calculate that for discrete traits, like in this case, but also for continuous traits. So in case of discrete traits, so here we have two traits, eye color and hair color. And we have two variants for each of them: blue eyes, brown eyes, blonde hair, and brown hair. So with these two traits and 4 combinations together, we can get 4 unique individuals. So we have basically 4 individual signatures. And when you do the binary logarithm, you will get like how many bits of information is needed to encode this information. So this is for discrete trait, but you can do very similar thing in continuous traits, and you do it in a way that you calculate the whole total variation, like you would calculate the entire acoustic space that is used by the species, and then you look on each individual and you see how much variation there is in each individual, and you compare the total variation to average within individual variation, and how many unique signatures you can find. So I hope this is a very basic introduction, but you can check the paper on the bottom of page. It's very well described, and I think the basic concept is very, very easy and very, very intuitive. So we got the tool which allowed us to make the comparisons, and then we started to ask which conditions promote acoustic individuality. So that means why some species have higher individuality, why in some situations we find higher individuality, and so on. And actually, I was talking about that we have many studies on many species, and this is big advantage of acoustic or acoustic domain, because we don't have so much material in other modalities. Like in visual, there is not so many studies focusing on individuality and quantifying individuality in visual and olfactory signals, for example. So in acoustics, we have many, so that's good for comparative studies. So we did the overview. We found over 250 studies. Then we did some filtering, but we still ended with over 200 studies, which concerned almost 200 different species. And if we look on which species were involved, it was about almost like 100 species for mammals and 100 species for birds, half to half. There are some there are some amphibians that were studied, about 10 different amphibian species, and also there are some reptiles and fish, but these are mostly 1 or 2 species. It's very rare. It's not that the individuality would be rare, but it's more about that the studies were not carried out, I would say. So here we can see how much individuality has been found for different species. So this is the scale on which we are now. So there actually are few studies that found almost no individuality. One of them is some Hawaiian monk seal in juveniles, and it's possible to say that they actually don't have individuality in their voices, or the authors didn't find it. On the other end of the scales, there are bottlenose dolphins. So these are that's the species that was found to have the highest individuality among animals. They are well known for their whistle signatures. So each individual develops the signature when they are young, and they use it to contact the others. They use it when they are alone and they want to join again the group, and so on. So in that case, so the Beecher statistics is almost 14, and it can give as much as 13,000 unique signatures. So it's remarkable signaling system. So somewhere in the middle, there is little owl. That's a species that I'm focusing on now. And there's not many studies on humans, but there are some. And for example, one study, this is probably the highest published for humans, but that's only concerning this sound, and like its harmonic structure formants, they carry about 6 bits of information and about 50 to 60 unique signatures. So then we have also other sounds which can also which can add up actually, but this kind of analysis hasn't been done yet. So now we have overview how much individuality we have in different species. So we can check how different species live. We can check different situations, and we can try to start to think about what is the driver for individuality. So which species make themselves more distinct, let's say, or whether there are some species in which it's important to make oneself distinct from others. So these studies were done previously. There are a few of them. Usually they are like focusing on a relatively small set of different species. And from the beginning, social group size and complexity are like one of the factors that might be important. So this is very nice study about on ground squirrels and marmots. And these are rodents living in groups, but different species live in different, have different group sizes, and basically different family size. And also they have, the researchers recorded alarm calls, which they do when they see something, for example, people or predators. They will start to cry, alarm calling, and the individuality in those alarm calls was positively correlated with the group size. So this is the evidence that group size probably drives. So the bigger the group, the higher individuality you need, because you just encounter more individuals on a daily basis. So another factor that might be important is mother-offspring caring strategy. And different species, so recognition of parent-offspring recognition, it's very important, because if you don't, so many species leave their offspring unattended for some periods of time. So if they are not able to reunite, then the offspring can die. And this is, of course, for parents, this is not the situation that is that should happen. So there are comparisons. So many species on individuality come from sea lions and seals. And there is some evidence that in sea lions, which are living in bigger colonies, they have longer period of lactation. They care more about offspring, and also they leave their offspring unattended quite often, and they don't use special places. So they, if the parents are not there, so the pups would intermingle and form something like crashes and groups of juveniles. So they definitely need, when parents come back, to recognize parents, and parents need to recognize their pups. So in sea lions, the individuality seems higher because of those reasons than in seals. So this is again to our research, and we didn't compare different species, but we focused on comparison between different situations. So we looked on calls that are given in different contexts, like sexual context, non-specific movement. Some species provide or give food calls when they find the food, distress calls, contact calls, alarm calls, agonistic calls, affiliative calls, advertisement calls, and echolocation calls. So we compared how is it in different situations, and in agreement with the previous studies, we found high individuality in contact calls. So that's when parents, for example, are looking for the youngs, they would do contact calls. Or when you are signature whistles of dolphins, that's also the contact calls, because when you get separated from the group, you start calling your contact call, and that is to re-establish the communication with your group. But and contact calls are like common and very important in group living animals. But on the other hand, we also find very high individuality, and even higher than in contact calls, in advertisement calls. And these are calls that animals make to defend their territories, and to attract mates, for example. And quite often that's solitary or pair living animals that do that. So that's not group living animals, definitely. And still we find the highest individuality in these kinds of calls. So this is something new. So we wanted to investigate more, and now we are asking why there is such a high individuality in territorial calls. So maybe one of the ideas is that when you do mistake, it's more costly than in group context, because when you are in group of cooperating individuals, you can maybe ask the other to tell it again, or if you don't hear properly for the first time, nothing important happens. But when you are in like competition situation, mistakes can be fatal, or more fatal than in group setting. So we are asking whether individuality depends on breeding density, and the logic is very similar to actually like the group context, because the more individuals you encounter, the more individuality in calls. Basically, we assume the more individuality is required. So this research has been done on little owls, and in Hungary. So there are still some little owls in Denmark. There's very few of them. The same or very similar situation is in Czechia, where we have about 100 last breeding pairs, but it's very different from Hungary. In Hungary, there are areas where they are very common, and we were able to find sites in which we find many males together. Usually this is happening in villages. We call them clumped males. And there's really little owls are calling the every 100 meters, for example, in villages. And then there can be 50 calling males in one quite small village, let's say of 2,000 inhabitants. And it's almost like all the population in Czechia. On the other hand, what we can find there also is isolated males. And these males, because little owls are now breeding mostly in human settlements, human buildings, houses, and so on. So we can find isolated farms, and each isolated farm will have one little owl and with no neighbors around, because there are no other houses around it. So you can find isolated males and clumped males. So the social environment of these males is very different in each case. And what we have found is that actually in clumped males, as we assumed, we found higher individuality in the calls. So larger variation, let's say they use different calls, territorial calls, which they use different melodies. And I have some example of this. So we are very happy to find such result. So here we didn't use Beecher's statistic because it's kind of population metric, but we rather used how much overlap there is in acoustic features between different males. So here it's a bit confusing because I say there is higher individuality in clump males, but the box plot is like close to 0, but that just means that there is less overlap in acoustic features between males, so they are more unique. So this is example. This is an example of like two males, two neighbors, and you could hear that each of the male is very consistent how he is hooting, while there's like very nice distinction between both calls, and you are easily able to follow which male is calling. So the last part, vocal individuality in humans and animals. So individual recognition in humans is probably very important for us And when people are listing what is unique to humans, this is not often mentioned, but probably people are one of the best, maybe the best individual recognizers in animal kingdom. So we see it how like individual recognition plays quite prominent role in some cultural rituals and beliefs. So, for example, carnival is a ritual that allows you to get rid of your identity for some time, some limited time, and do things anonymously. So I don't know the wedding rituals here, but in Czechia, one of the wedding rituals is recognizing your wife by the shape of the legs, and of course also like the rituals and beliefs. So like when we turn to beliefs, we put a lot of we somehow tend to think that if two people are very similar, they are somehow related to each other. So that's the example with Kieślowski film La double vie de Véronique, and also if you if you look at social behavior, social organization, very likely whether you stress individual traits or whether you suppress them, that has some consequences on how the social society is organized and how it works. So humans recognize mates by smell. So this is an example of the fairy tale situation in which the prince has to choose a princess, his beloved princess, but actually the princess is among two other lookalikes and they cannot speak. So we have visual signal blocked, or visual signal doesn't give you any information. Acoustic signal doesn't give you any information. So the only one that is left is smell. So people know that smell is important for individual recognition for a long time. But of course, faces play a prominent role, and there are nice papers showing that face holds lots of variation and independent variation. It's not just that some dimensions increase, and also genomic parts or gene parts that encode the variation in face are more variable than those that encode variation in other body parts. Again, some examples from the culture. So I very much like this monodramatic series from Daisuke Takakura. So if you look on these pictures where everyone looks the same, it's kind of weird feeling. Some photographers focus on taking photos of twins. So twins, yeah, it's interesting topic, whether they are different, whether they are same. This is also a nice series of pictures focusing on 100 years old people and comparing them with photographs when they were young. So here you can compare which traits remain stable for a long time period. And from art, for example, when you like team that makes some art disturbing, is when you use no faces, or where you somehow destroy the faces. It's kind of disturbing on paintings. So in human voice, individuality is encoded by many different features, and linguistic features, but also non-linguistic features resulting from vocal tract. I think I could, for any one of them, I could give you examples how this is done in animal kingdom as well. But what is different? So we as humans, we have tendency to see the individual recognition ability as something very natural, but that's not true for many animals, and they don't need some animals don't need to recognize so many individuals as us. So sometimes they do just recognition my offspring versus all other offspring, but they don't, for example, recognize different individuals within offspring. So nine birds in the nest, for the parents, it doesn't, it's not, they are all equal. So they don't make distinction for individuals in their group. Sometimes, so I already mentioned Hawaiian monk seal example. So not only that they don't have much individuality in their voices, but very likely they don't care about individual recognition. So the pups, they just move around the group and they suckle from other, from different mothers, and the mothers don't seem to recognize or don't seem to care about individuality of the pups. So I'm coming back to elephant example. So those studies on elephants in general found very low individuality, but at the same time, we know that they are able to recognize many individuals, and they are long-lived animals. They keep the groups stable, but also they travel among different smaller groups. So it's quite complex, quite large community, and they need to recognize many individuals. And we don't know how they can do it with such a small individuality in the rumble calls. So these are corn crakes. This is a different example, and it has been found that there is high individuality in their calls, maybe 1,000 unique. The variation or individuality is so high that it allows encoding of 1,000 unique signatures. We know that they use it for neighbor-stranger discrimination, so they are able to recognize different individuals. They also use it to, so if one individual hears the contest of two different individuals, to other individuals, then he listens what happens, who is the winner, and then he acts accordingly to winners and losers of those fights. So they can eavesdrop and use individual recognition for eavesdropping. But so when researchers try to test for like what they thought that is the trait responsible for that recognition, they were not successful. And maybe so they tested the pattern. So actually each is composed from unique sub elements, and the intervals between them are very characteristic. But maybe this is too much detail. Maybe that's something like our fingerprints. So we know that fingerprints are unique, but we don't use them in daily life to identify others. So it's good to know also that sometimes there's different vocal production mechanisms. So for example, birds, actually some birds have two sound sources, and they can use each part of the vocal tract to generate two tones, which are very close with the frequency. And this is then like these two tones eventually, like when paired together, they go into pattern, they make a beat. So it's woo, woo, woo. And the beat is characteristic for each individual. And now I'm coming back to chiffchaffs, because so birds are also, they produce sounds very efficiently, and basically they tune the vocal tract to they tune the vocal tract to the fundamental. So there's there's not much in harmonic, in voice quality and harmonic harmonic frequencies, formants in their voice. So again, this is chiff chaff. You can listen to them when you go outside today. So they are, it's very easy to recognize. They just do chiff chaff, chiff chaff, chiff chaff all the time, just two different sounds. So we tried to find what is individual distinct in those chiff chaffs. And what we found was that actually those two sounds are not two sounds, that they make many different syllables. They just sound very similar to our ears. But when you look on the spectrograms, that's different syllables. So we tried to make a repertoire, which maybe for some animals, when you do the repertoire of syllables, you will find like which syllables they use that's characteristic for each individual. But we were not able to construct repertoire because every now and then, after 1,000 songs, they still were able to switch to another set of chiffs and chaffs. And we couldn't compare even chaffs in between different birds and so on. So we failed to find the individuality feature in chiff chaffs, but we still know that they can recognize neighbors based on single random song. So we unfortunately found that songs are constantly changing and don't appear to have individual features. So for us, it's mysterious how they do it. And we also tested some deep learning methods, and they seem to pick up some short-term cues about individuality. But when we tried to recognize the same individuals after 1 year, we failed. So, take-home summary. I wanted to show that animal vocalizations in general carry individually specific features. We can measure it and we can compare it, and we should do that because it can answer some nice scientific questions. We can learn much more about vocal communication and individual recognition. And I hope that I showed some important and interesting differences between humans and animals, and that I gave you some overview how these things might be studied in animals. And that's it. This is acknowledgement. I worked with many colleagues, and I would like to thank to all of them. And I know that I was long, and it's already time to go for many of you. So I apologize for that, but if you have questions, I will be happy to take them.
Thank you so much.